Bienvenidas y bienvenidos a este seminario del Instituto Nacional de Salud Pública, un espacio que nos convoca para reflexionar, compartir conocimientos y fortalecer nuestras acciones en favor de la salud pública. Es un privilegio contar con su presencia y participación. Con ello, damos formalmente inicio a nuestro programa.
Bienvenidos todos y todas al Ciclo de Seminarios de Investigación y Control de Cáncer del Centro de Investigación en Salud Poblacional del Instituto Nacional de Salud Pública. Es un gusto tenerlos a todos con nosotros. El día de hoy tenemos la participación de dos muy destacadas científicas, la doctora Sabina Rinaldi, que trabaja en la Agencia Internacional de Investigación en Cáncer. Ella es la co-líder del equipo de hormonas y metabolismo y también es la Deputy Branch Head de el Nutrition and Metabolism Branch. Ella es originalmente química de formación y recibió un doctorado en epidemiología en la Universidad de Utrecht en Holanda. Ella ha trabajado en la YARC desde 1998 y su carrera se ha enfocado en cánceres relacionados a hormonas, específicamente cáncer de mama y cáncer de tiroides. Como comentarista tendremos a la doctora Gloria Sánchez, que es profesora de investigación de implementación en la Escuela de Salud Pública y coordinadora de investigación en infecciones y cáncer en la Escuela de Medicina de la Universidad de Antioquia en Colombia. La doctora Sabina nos hablará del estudio PRECAMA y la doctora Gloria Sánchez es colaboradora de este proyecto, entonces ella será comentarista de la presentación. Iniciaremos con la presentación con la doctora Rinaldi y posteriormente la doctora Gloria Sánchez dará unas palabras de comentario y luego abriremos a preguntas. Doctora Rinaldi, bienvenida.
Yes, muchas gracias. I'm very sorry. I need to switch to English because my Spanish is not really good. So the presentation is going to be in English. So thanks a lot. Thanks for the invitation to be part of these seminars. I'm going to share my presentation. Let's see if I manage. Can you see this? I hope you can see my
Sí, se ve
slides.
bien. Sí, se ve bien.
Okay, thank you very much. So thanks a lot, really. It's a real pleasure to be here and to present the PRECAMA study here to you. I hope that sometimes I'll be able to come physically to your institute later on in the future. So this is going to be a short presentation. So on the PRECAMA study, I will introduce you why we were interested in setting up this study in Latin America, and then I'll present some of the results that are coming out of the project. So, okay, let's see if I manage to show the slides now. Yes. So what is going on in Latin America now? Well, as you can see here from these slides, there are some demographic changes that are not going on in the continent. The continent is in a transition stage with some demographic changes. As you can see here, this is the population distribution in the 90s. So you can see that now this is the distribution in 2020. You can see the population is now really aging much more than before, and the population is also growing and getting older, and these changes are accompanied by other different changes in the population. There are changes in demographic and sedentary lifestyle, changes in dietary habits, changing in smoking and alcohol intakes, alcohol consumption, also changes in some environmental carcinogenic exposures, and a lot of urbanization. So this is really a population in transition. So what is this transition that is going on, and how this is reflected into health, let's say, for for the continent. This is translated into changes in the percentages of communicable versus non-communicable disease on overall diseases. As you can see, in the early 50s, most of the diseases happen in Latin America were communicable diseases, and then the trend is really in lowering the percentage of these communicable diseases. This is due to better advances in medicine and in science, of course, but in parallel, you can also see in the, I'm not sure I have the pointer, but you can see in the red part that actually non-communicable disease part is really getting much more prevalent. Non-communicable disease, they include cancer, cardiovascular disease, and diabetes that are now leading causes of disease in Latin America, and they are responsible for about 80% of deaths in the population. So my talk is going to be more focused on cancer. When we look at the leading causes of cancer, meaning cancer incidence and cancer mortality in women in Latin America, you can see on the left-hand graphic here, picture here, that basically breast cancer is the leading cancer in Latin America in almost all the countries here. For mortality, still the leading. There is some part of the continent in which still cervical cancer is the leading cause of mortality, some lung, but actually most of the continent is really the mortality. Breast cancer is really number one mortality among women. When we talk about rates of incidence and mortality in women, these are the recent data from Globocan and the agency at IARC. You can see on the left-hand side, this is incidence throughout Latin America. And at the bottom, you can see the standardized age standardized rate of the United States and Spain, just for comparison. As you can see, some of the countries, like for example, Mexico, the incidence is really lower than what we see in Spain and the United States. But then if you switch to the right part of the graph here, where we see mortality, actually, this is still comparable, actually, what the mortality rates that we have in the United States and in Spain, meaning that still mortality is really a concern in this population. This is also confirmed here. This is the graph of mortality. That you can see here on the right-hand side. Most of the countries in Latin America, and you can see Brazil, Colombia, Costa Rica, and Mexico, especially here, you can see there's in some of these countries, the rate of mortality is really still increasing, and this is of course a concern. This is due to many different reasons, including probably access to care, but also some of the changes actually that we discussed before. And what is important about this increase in terms of mortality is that it becomes really important to prevent the disease, because once you prevent the disease, basically, you don't have it. So the probability of dying for it is actually really, it doesn't exist. So that's why we're really focusing on prevention. When we talk about projections, what's going to happen over the next years, because those were data from 2020. If we see over the next 20 years, you can see that actually the overall burden of cancer is going to increase, and it's going to be more than 60% actually, from 50 to, oops, sorry. I don't know what happened. Sorry. I hope you can see my slides again. Sorry.
Amplifícalas. Just make, exactly, that okay.
Is it okay? Yeah, I'm sorry, I don't know. So as you can see, okay, you can get between 50 and 70% increase actually of cancer, stressing the importance really of working on cancer prevention in this particular region. In addition to increasing incidence and still increasing mortality in some of the countries, There are also differences among the different types of cancers that we can observe in Latin America compared to Western countries or the United States, is that actually we see that the incidence of breast cancer in young women is still quite high, is almost double than what we see in the United States. So you see here is about 20%. When we talk about young women or early onset, we talk about cancers before age 45. As you can see here on the left-hand side, it's about 20% in Latin America, while we talk about less than 12% in North America. And this is really very important because, of course, this affects a very young population with all the concerns that you have in, of course, getting cancer in general, but especially in young women, where you can have career disruption, family care disruption, pregnancy concerns in addition because of the treatment, fertility preservation, lots of additional complication actually is an incredible burden on these women and also on society. So what do we know about risk factors for breast cancer? Well, especially in young women, because this is what we are going to focus on today. Well, there are a few risk factors that are already known. Actually, this is not, oh gosh, sorry. These are only some of the risk factors that are known. Some of them are modifiable. As you can see here, you can see some reproductive variables like pregnancy and breastfeeding that have been associated to a decreased risk in premenopausal women, but also some factors that can increase the risk of breast cancers like smoking intake and alcohol, physical activity. Body mass index has been a little bit of a complicated association that we see in premenopausal women. Many studies reported an inverse association that is still not really well understood. And there's also a part of genetics. That is actually more important in premenopausal women common than postmenopausal breast cancer. So there's really a paucity of study investigating risk factors. Some of these factors, like the genetics, for example, are not modifiable. What we are interested in are risk factors that can be modifiable. Because if we identify modifiable risk factors, then we can maybe act on the risk factors, trying to lower the incidence and prevent risk. And this is really what we are focusing on today. So this is the report of the World Cancer Research Fund and the American Institute for Cancer Research. That's the latest global report that was published some years ago. There will be a new one soon. As you can see, they focus more on diet, nutrition, physical activity, and you can see the evidence here of the association in premenopausal women. As you can see, the convincing evidence of factors that are associated convincingly in premenopausal women is not too big. You can see also, you can only see one factor that has been associated to in increasing breast cancer risk, which is height. And then you can see still what is associated to physical activity, but also body fatness and other factors that we mentioned before. The evidence is still probable, but not convincing. Let's see what will come in the next report. And all that is associated to diet and other types of different environmental exposure is still limited and suggested as an association on as definition of risk for breast cancer. What we see in postmenopausal women is a little bit more, there are more factors that have been classified as convincing evidence with the increased increase in risk for breast cancer. You can see alcoholic drinks here on the right-hand side, body fatness, adult weight, and attained height, and still some probable evidence of physical activity. And that would be reclassified, as I think, with the next classification. There have been some, well, most of the data are actually coming from the WCRF and the ICR report are coming from cohort studies that have been developed either in the United States or in Europe, but there have been also some studies that have been undertaken really in Latin American women living in Latin America. And you can see that here we see some of the risk factors that we saw before that were already investigated in Latin America, but most of the studies had small sample size, and especially when we investigate the association in premenopausal women. So there are some reproductive factors that have been identified as associated as protective for breast cancer in premenopausal women. Physical activity as protective, alcohol intake, as we saw before, again, even in Latin America as increased risk. Obesity, where associations were a little bit less evident, both pre and postmenopausal women. Concerning diet, again, which is something that is a lifestyle variable that can be modified. You can see the intakes of carbohydrates and red meat has been associated with increased risk, and Western diet as well. The increase of breast cancer in sugar sweetened beverage consumption, protection from vitamin D, and family history of breast cancer has been increased the risk, even in population in Latin America, and these factors are quite similar to what's been observed so far in other populations. But what is going on now? We talked about these changes in the population. So there are, as I mentioned already, some lifestyle changes that are occurring in Latin America. These are a couple of examples that we can see, that we can have a better idea of what is going on actually in Latin America. So, for example, we talked about obesity and the association with breast cancer. As you can see here, there has been quite a substantial change actually in obesity over Central Andean and Southern Latin America between the 80s and 2010. And you can see that basically the percentage of BMI lower than 25 has been squeezing over this time period, and then mean BMI has been really increasing over the last years, having a big switch for a mean BMI of 23 in the 80s to a BMI of 27 in the years 2000. Some other changes that have occurred in Latin America, as just examples again of this epidemiological transition that we discussed before. This is, for example, changes in the intake of ultra-processed foods in Latin America. We will talk about this a little bit later on. As you can see, most of the actually Latin American countries has been an incredible increase, tremendous increase in the intake of these processed foods, as we call them. And in most of the Latin American countries, there's been quite a steep increase of the last years. So there are many things happening and going on in Latin America over the last 10, 20 years. So what we thought would be really of help to understand a little bit what is happening and if these changes can be associated or can have an influence on the different types of cancers that we discussed before. So we decided to set up a multicentric study, which is called the PRECAMA study, molecular subtype of premenopausal breast cancer in young Latin American women. And it's a multicentric study, yes, that we undertook in 5 different countries in Latin America, really targeting young women who were between 20 and 45 years of age. So we started the recruitment. It's a population-based case control study that's been undertaken in 5 different countries with the help of all the PIs and the coordinators locally, of course. It's invaluable help to set this up. Dr. Gabriela Torres in Mexico, Dr. Gloria Sanchez in Colombia, Dr. Carolina Porras in Costa Rica, Dr. Fabiana Vasquez in Brazil, and Dr. Maria Luisa Gramendia in Chile. So we recruited a total of about more than 700 cases and 700 controls. We defined the cases as I said, women that developed, they had breast cancer, and they were diagnosed with breast cancer. They had positive biopsies and tumor staging for the cancers that were identified, and they were aged between, as I said, 20 and 45, and they were living in the same area for at least 3 years. We recruited the cases before any therapies, and to these cases that were recruited, we also recruited some controls. The controls were matched to the cases. They were recruited through the general population in most of the countries, and they were matched to the cases on age, plus or minus 3 years. They should have been living in the same district as the case for at least 3 years. And in some of the centers, they were also matched on to the cases on Social Security when this was important. During the recruitment, these cases actually they were recruited through different hospitals and private oncology institutes that were providing care to a large population and urban population, especially with a wide range of socioeconomic status. And why these subjects were recruited, they were interviewed by nurses, and they had to answer many, many questions on lifestyle, social economic status, current social economic status and during infancy, for example, reproductive history, reproductive health, health in general, lifestyle. They had anthropometry that was measured, and in Brazil, actually, they had anthropometry measured in a standardized way, and they measured, height, waist and hip circumferences. That is what is usually collected for this kind of studies to see the association between anthropometry and cancer. But also in Brazil, they had the possibility of measuring impedance, so that you can have better understand the percentage of body fat and of lean fat tissue, or lean tissue actually of a person, more than just measuring the usual anthropometric values. We also collected a lot of information about the environment and, in a general sense, physical activity as well, use of hormones, medical history of cancer, and also there were administered some full frequency questionnaires to better understand what this population was eating. And these questionnaires have been validated locally, and they were really specific of the population of each of the different countries where the subjects were recruited. In addition to the questionnaire data that we recorded, we also collected biological specimens because we were really interested in analyzing a mechanism in molecular epidemiology, as we say. So we collected a kind of a biobank and biological specimens. Some of the specimens are stored locally. The other half have been centralized at the agency to run general analysis on all the PRECAMA study with all the different countries. So we collected plasma, serum, red blood cells, buffy coats, and also some spot urine samples that were stored at -80. And in addition, since this is a case control study, we had the possibility of also collecting tumors from the biopsies or from the tumors when they were going through the surgical intervention, but before any adjuvant therapy. The tumors that were collected did not have any intervention on them, and we had some cuts of these tissues for immunohistochemistry analysis that were shipped to a centralized lab in the United States, the lab of the Fortalecer Cancer Research Center led by Dr. Peggy Porter. And also we also had some thicker cuts that were cut and sent to the same lab to be able to extract DNA and do some genomic analysis that were sent again to the Fortalecer Cancer Research Center. Since this was a multi-center study, we thought it was really important to have standardization of the immunohistochemistry analysis. That's why the analyses were done at the centralized lab. But we also organized some meetings to discuss the standardization of pathology, standardization of biomarker testing. We had 4 different meetings. Thanks to the help of UICC and CTED, so that we made sure that the standardization of the PRECAMA immunohistochemistry analysis and pathology, and develop standards for tissue sample processing, immunohistochemistry reagent, and interpretation of the findings, pathology review criteria, and improve the local facilities and diagnostic capabilities. As you can see here, the workshops that were organized by Dr. Peggy Porter. We also had some additional workshops, always with the help of CTED, to discuss more topics that were more associated to the etiology of breast cancer, like nutritional epidemiology, molecular epidemiology, and of course genetic counseling, because as we said before, there's a higher part in any case of high-penetrance mutation genes in this type of early onset of cancer compared to postmenopausal women. So let's go to the results part. Sorry, those are a little bit busy slides, but I'll guide you through all of them. Just mentioning some of the results that we observed. You will see during the presentation that the number of cases and controls that were included in the different studies are a little bit different, because we analyzed the data, investigated, ran our investigations when the recruitment was still ongoing. So that's you will see that for some of the earlier publication, let's say the number of cases and controls included in the studies is a little bit lower. And then for those published later, we had more cases and controls. The other thing that I wanted to say is that for the time being, Brazil was not included in the analysis that we did, because they joined PRECAMA a little bit later. Everything started in 2012, but Brazil joined in 2018, I think. But that will be included in the next round of publications. So as you can see here, very briefly, this is description of cases and controls. Well, the mean age was about 40 years old. Cases were had more children. Controls had more children than the cases. Cases were older at the first full-time pregnancy. As you can see, I don't know if you see my pointer, but in any case, I'll just go through the different significant associations, significant variables between cases and controls. And they had, and they breastfed less, actually, cases, as you can see here. Oops, sorry. They didn't have differences, actually, family history of breast cancer, nor in alcohol intake. Cases were nevertheless less active, they had less physical activity. And there were cases where they had lower BMI, and they were a little bit taller than the controls, and they weighted a bit less. Just an overall description of the tumors, the type of tumors that we have. Again, you can see that there are only 332 cases included in this analysis. Those were the cases for which we had decentralized immunohistochemistry done. The others also have the subtype. We need to keep going with the centralization of the results of the immunohistochemistry. But for the time being, for the 332 that we have, as expected, most of them are estrogen receptor positive, progesterone receptor positive, about 70% HER2 positive or 15%, and we had only about 21% of triple negative breast cancer, and most of them were basal-like. Just some analysis, some results. Actually, on the first analysis that we ran, we mentioned reproductive factors and breast cancer and the associations that were already observed in other population with the risk of breast cancer. So we decided to investigate these reproductive factors in our population and see what the associations were. And sorry, this is a little bit blurred, but again, I'll drive you through this. When we ran these conditional logistic regressions adjusted for all the relevant confounders, we saw an inverse association with age at menarche, an inverse association with time since last birth. Parity also resulted as being protective on breast cancer risk, and breastfeeding as well. Age at the first full-time pregnancy was actually directly associated with breast cancer risk, as the longer time between menarche and the first pregnancy. And age at last pregnancy as well increased the risk of developing breast cancer in this population. Since we had the possibility of having all the information on the tumors, we stratified the analysis, wanted to see whether there were any differences in estrogen receptor positive, estrogen receptor negative, or triple negative cancers. And actually, most of the associations that we saw before, so pregnancy, time since last birth, parity, and breastfeeding, was indeed protective for estrogen receptor positive cancers. And age of full-time pregnancy, and the time between menarche and first full-time pregnancy, and age of last pregnancy was directly associated to breast cancer risk in estrogen receptor positive. In estrogen receptor negative cases only, we saw an inverse association, significant association only with age at menarche and duration of breastfeeding. And this inverse association with age at menarche was confirmed also when examining also only a triple negative breast cancer cases in this population. So we saw some of they can confirm some of the associations and on these variables, and breast cancer risk and the reproductive variable do play a role in this population. We mentioned the association of obesity with breast cancer risk. So we wanted to explore again the association between anthropometry. I told you we collected information about height and all the waist and hip circumferences and weight, and they've been standardized throughout all the centers. And as you can see, mean BMI in the populations were about 29 in controls and 27, in the cases. And then, as you can see in the small part here in the bottom of the slide, squares, sorry, you see that the percentage of women defined as obese with BMI above 30 actually was much more important in controls than in cases. So when we ran, again, conditional logistic regressions on this data, actually we saw an inverse association, which was quite significant. Which was significant and quite impressive, quite low actually, between BMI and risk of breast cancer in young women, while we didn't see increase with height. Waist and hip circumferences, they were significantly associated with the decrease in risk in continuous variables, but not in tertile variables. And so we saw so in other population, this inverse association between BMI and overweight with breast cancer in young women. We also collected information about what we call the body shape during life. So we had these 6 silhouettes here representing body shape, and women were asked to identify the body shape between different types of, between different ages, between 6 and 11, 12 and 18, 19 and 25, and then up to 1 year before the interview. And this is what we observed in the PRECAMA population. I'm not sure you can see it, but the most frequent silhouette that was identified in childhood, in adolescence, and until age, it's number 3, until age 25, it was actually silhouette 1. And then the more women were growing up, let's say, then the silhouette 3 was chosen for later ages. So there was a change in the distribution. We also tried to identify some trajectories of what happened during early life, because we were interested in trying to understand whether increasing or decreasing weight or having being overweight during childhood and adulthood could have an influence that on breast cancer risk that is different from the adult one. We identified 3 different trajectories, actually looking at the different silhouette, and we identified 2 trajectories, T1 and T3 here, which is the top and the middle one, actually, that were named constant increase or increase at puberty for T1, and then we had a separate silhouette that was constant increase from being constantly lean, sorry, which is the trajectory number 2. We wanted to investigate the association of the trajectories with risk, and that actually we did not really see any associations of these trajectories with breast cancer risk. We simply saw an adverse association of the silhouette between ages 26 and silhouette at interview that basically correspond to the analysis on BMI that I just showed. We also started looking at the association between food intake. I mentioned the increase in ultra-processed food in Latin America. So we decided to investigate the association between ultra-processed food in breast cancer. So what are ultra-processed food? Actually, the term ultra-processed food refers to the processing of substances derived from foods like baking, frying, hydrogenation and hydrolysis, and they generally include a large number of additives and other types of the things that are used to conserve basically the food over time. The aim of this ultra-processed food is to create a durable, convenient, and also palatable food that can stay, that can be eaten readily, and that are ready to eat also, and that can be consumed relatively easily, and that do not really require a lot of process. So to be able to identify these ultra-processed foods, the NOVA classification has been set up by Montero and colleagues, and there are 4 types of the foods can be divided into 4 different types. NOVA 1 is contains the unprocessed, naturally minimally processed foods, like fresh fruits, vegetables, like unprocessed meat, fish. NOVA 2 is contains the processed culinary ingredients, many, rose butter, whatever you add to your recipes. NOVA 3, it includes the processed foods that usually put together NOVA 1 and NOVA 2, so the way you cook your foods, like canned fish, but also canned fish, sorry, cheeses and bread, for example. And NOVA 4 is really what we call the ultra-processed foods, so what I mentioned before, and usually what you see in ultra-processed food is soft drinks, sweets, savory packaged snacks, processed meat, and all this kind of ultra-processed food that is eaten. Actually, what we really wanted to explore more was the association of NOVA 4 foods in the etiology of breast cancer. So we started looked at started calculating ultra-processed food consumption from the questionnaire, frequency questionnaires that we had. On the left is the type of foods that were contributed in ultra-processed foods in PRECAMA, in most of the centers that were more or less the same, maybe with different actually percentages, but you have ready-to-eat heat foods, cakes and desserts as top 2 ultra-processed food consumed, carbonated beverages, breakfast cereals, sausages, yogurt, and dairy products. On the right hand side, what you have the distribution of cases and controls that are quite similar actually in these categories of foods. When we looked at the association again on between ultra-processed food and breast cancer risk in this population, we used 2 models. One is the model that is usually adjusted for all confounding variables, and model 2 actually is model 1, but adjusted also for NOVA 1, NOVA 2, and NOVA 3 foods, because of course if you eat more of NOVA 4, you eat less of the others. So we wanted to make sure that we were really comparing the results correctly. And as you can see, by running this conditional logistic regression analysis, we saw a direct association actually between the consumption of an increase in the consumption of ultra-processed foods and the risk of breast cancer in the population, which was mainly associated to estrogen receptor positive, actually breast cancer. We don't see a significant increase in estrogen receptor negative breast cancer. Sorry.
Sabina, I just want to let you know that we need to start to wrap up so that Gloria has some time to comment, okay?
Sorry. Okay, so we just had also some analysis of biomarkers that we investigated. So we saw that because we collected these biological samples in PRECAMA, and then we used some of them to look at the association between inflammation and breast cancer, and we saw an increase for some of the cytokines actually and breast cancer risk. We also had some targeted sequences in tumors. This was just to show it's a feasibility study to show that actually the tumors that we have in PRECAMA can be used for this kind of analysis. So they're in FFP blocks. We tested some breast cancer genes, and actually we saw some of the mutation in these genes, TP53 and PIK3, for example, AKT1. And actually we saw that the mutations that were more the same that can be found in other populations that we see in metabolic and TCGA, which are European populations. But actually, when you look at the mutational patterns with this gene, the gene of TP53, actually, the type of mutation that were different in these tumors. And this let us think that maybe there are really some local exposures that we still need to understand that can be associated to, that can lead to the development of cancer that may be different from other populations. So, in brief, apologize for being a bit late. We started investigating reproductive factors, anthropometry, processed food, inflammation. We also observed some specific mutations in tumors. There are already some ongoing projects that we still need to investigate, and we would like to investigate in PRECAMA, the role of endogenous hormones, to see whether we can find the same association as in other populations, fatty acids. We also started looking at metabolomics data to be able to have maybe new hypotheses on or identify new breast cancer risk factors. Some of the ongoing work on questionnaire data include the use of emergency contraceptive pills that is currently ongoing, sleep duration. So we're also interested in studying chronic stress and different nutrients actually that have been collected through the use of the full frequency questionnaires. PRECAMA is also part of the Conference project, which is the largest project in the world on breast cancer genetics that is coordinated by the NCI in the United States, and to look at the association between genetics and breast cancer risk. And we will soon hopefully receive the data from the conference to work on precisely on the PRECAMA study. The next steps, I told you about the inclusion of Brazil and the use of their data, standardization of the procedures so that we can increase the number of cancers with receptor status have been centralized. I think it would be nice if we could eventually have the data for survival analysis. We could also work on survival and not only on risk, but of course this requires funds and support. We'd be interested in looking at obesity phenotypes to better understand why obesity is in BMI, let's say, is associated to a decrease in risk. We also know that the population in Latin America is there's a lot of that mixture, so we would like to get to be able to identify and to work on that mixture and see whether the association with breast cancer differ by stratifying the population. Be very interested in working on proteomics as well, doing more than only targeted inflammation, see whether there are some other proteins that can be involved in cancer development. And then, well, all the aspects, let's say, more on environmental exposures like pollutants, pesticides that are maybe less strongly associated in the literature than to other cancers, I mean, to breast cancer than other cancers, but I think that exposure maybe in earlier life can still play an important part. And the microbiome as well, that can also be an interesting new way to better understand breast cancer in young women. So that's all I wanted to say. I simply wanted to take a couple of minutes more to thank, because I've been coordinating this over the last few years, but actually this has been an incredible amount of work from all those the colleagues who were involved locally and all the teams, so not only the PIs, of course, and but also the teams in the different hospitals, everybody, a lot of work. It was incredible and substantial, really, to get this going, and I will never stop thanking this, the contribution of everybody. And especially, I mean, the study has been really started by Dr. Isabella Romieux, so I'm just following up her coordination, actually, and there's been also a lot of people at the agency trying to work on this, and of course the contribution of the team in Seattle, the Fred Hutch Cancer Center, and I thank you very much, all of you, for listening to me, and I'm sorry I'm a bit late.
Hi, thank you very much. Hola, Gloria. Ya te escucho.
Hola. Bueno, lo hago en español. Voy a hablar despacio. Or you want me to speak in English?
You can speak in Spanish, and if I don't understand, I'll let you know. But I'll answer in English because, sorry.
No, no te vemos, no te vemos. No sé si apagaste tu cámara o...
Aquí estoy. Sí, la había bajado, pero aquí estoy. Bueno, la doctora Sabina nos acaba de presentar un estudio de casos y controles altamente riguroso en todos sus aspectos metodológicos, y esa es como la fortaleza más grande que tiene este estudio, un esfuerzo muy grande en toda Latinoamérica, o por lo menos en países con unas poblaciones muy representativas, de una población que es muy mixta cultural y genéticamente y étnicamente. Además de eso, todas las mediciones fueron muy rigurosas, y eso pues da mucha validez a los datos que ella presenta. Algo que contribuye muchísimo al conocimiento es precisamente que es la población premenopáusica o mujeres menores de 50 años. Esto tiene dos cosas muy importantes. Uno, porque hoy en día no tenemos programas de prevención en las mujeres menores de 50 años, solo hacemos la tamización en Latinoamérica en mujeres mayores de 50 años. Entonces, estos datos van a proveer precisamente, o están proveyendo, la fuente de información para hacer prevención primaria en las mujeres menores de 50 años. Y de hecho, estos datos alimentaron mucho el Código Latinoamericano de la Prevención del Cáncer, ¿no? Los cuales incluyen, por supuesto, el uso del alcohol, también la actividad física, los alimentos procesados, como factores de riesgo comprobados y confirmados, y que si hacemos intervenciones específicas para dar respuesta a estos estilos de vida, ya sabemos que tienen un efecto impresionante en la reducción de la incidencia de cáncer de seno y otros cánceres. Entonces allí hay una fuente de datos supremamente importante. El otro punto que yo quiero resaltar de este estudio es la parte relevante de visibilizar todos y cada uno de los diferentes estilos o comportamientos que tienen que ver con la salud sexual y reproductiva de la mujer en Latinoamérica. Algo que no es muy visibilizado, no es informado, muchas mujeres no conocen, y que inclusive tuvieran alternativas para bajar la exposición a los estrógenos, si tuvieran esta información a la mano. Entonces yo creo que ahí también hay mucha oportunidad de la información que nos arroja ese estudio, de cómo diseñamos intervenciones para que las mujeres puedan tomar opciones verdaderamente no utilizar solamente algunos métodos que son los que incrementan los niveles de estrógenos, o partir los tiempos, o incrementar la actividad física si están tomando los anticonceptivos hormonales, etcétera. Entonces, todo eso es supremamente importante para visibilizar en todas las poblaciones del mundo, pero especialmente esos datos no existían en la población latinoamericana. Me quiero referir a lo que yo he visto y que considero muy novedoso de la investigación, es el biobanco tan impresionante que tiene este estudio. Ese biobanco ya empezó a arrojar resultados muy importantes, y me voy a referir específicamente a las citoquinas inflamatorias y su relación con el uso de los estrógenos. Esto es supremamente importante, en donde nos estamos preguntando si el tamoxifen puede ser una medida preventiva en cáncer de seno, y sobre todo para este grupo de edad, menores de 50, en donde no tenemos mayores medidas preventivas, ¿sí? Otros datos que son muy interesantes y muy útiles de esta investigación, dado el rigor científico con los que se adquirieron, es cómo estos datos pueden informar los modelos de riesgo que se están desarrollando en todo el mundo. Inclusive el hecho de estar participando en el PRECAMA, vamos a conocer la gran cantidad de factores genéticos asociados a los cánceres en mujeres más jóvenes de la región, para poder diseñar programas de lo que llamamos el cáncer de seno familiar con esta información. Y también tener los scores poligénicos de riesgo, que nos ayuden a integrarlos en los modelos de riesgo que se estén desarrollando en esta población, para evaluarlos el día de mañana, para estratificar la tamización. Entonces yo considero pues que este estudio es verdaderamente un avance importante en el conocimiento de cáncer de mama, que no lo había en la región, tan completo, tan riguroso, con un emparejamiento con controles por estrato socioeconómico o por hospital, que es uno de los problemas que a veces, y las limitaciones que tenemos para hacer los estudios de casos y controles, la rigurosidad de la confirmación del caso incidente, eso es supremamente importante en este estudio, la inmunohistoquímica, o sea, todos y cada uno de los detalles nos dan la confianza que la evidencia es muy sólida, y que esto va a contribuir definitivamente al bulto de evidencia que se tiene que generar para tomar decisiones, para crear intervenciones que tengan un efecto en el cáncer de mama en la región. No sé si tengan alguna pregunta para la doctora Sabina o...
Sí, quisiera, digo, ya nos empezaron a llegar preguntas, pero quisiera empezar con una pregunta muy general. Hemos escuchado mucho recientemente sobre, digamos, reclasificar no nada más cáncer de mama, pero muchos cánceres como cánceres de inicio temprano. En el caso específico de cáncer de mama, se utiliza dependiendo, pero a veces hace menos de 50 años, o menos de 45, inclusive menos de 40 años. Entonces, como que quería entender, es una pregunta para ambas. O sea, ¿cuál es la opinión respecto a esta nueva forma de entender el cáncer y cómo, digamos, esta clasificación por más bien por edad que por estatus menopáusico puede cambiar o no la forma en la que interpretamos la información? Y si crees y cuál es su opinión sobre, pues, empezar a realmente, en vez de estar haciendo análisis en pre y postmenopausia, sino en cáncer con las diferentes definiciones que ya mencioné, de inicio temprano versus inicio posterior.
¿Quieres contestar tú, Sabina, o
In English.
in
Okay,
English? I'm
fine.
sorry.
I'm sorry.
Yes,
I
okay.
don't manage Spanish.
That's okay. I
Did
can
you
repeat
understand
the
my question?
No,
but
I understood about, yeah, early onset. That's the question about
early
early
onset.
onset. Yeah,
Exactly, early
well,
onset.
let's say that the way that the PRECAMA women were recruited is mainly early onset, right? Because it's less than 45. So basically it's the
Okay.
same. It's not premenopausal women, but less than 45. So that's been really focused on, let's say, young premenopausal breast cancer, what we called at that time, but actually now it can be defined as early onset.
Okay.
So that's now let's see actually what happens, because there's still an early onset increase of premenopause of early onset breast cancer, let's say, that is really happening in different parts of the world. This is also something that we do not understand, but I think that PRECAMA can really be a very important study that can really help answer some of the questions actually on early onset, because having more than 700 early onset breast cancer cases is really a precious study that we can really use, and we should really use to address some of the important questions about why now we have younger women getting breast cancer compared to what happened some decades ago.
So, on a related question, oh, sorry, Gloria,
Sí,
adelante.
yo quisiera agregar ahí que hasta ahora solamente se han estudiado los factores clásicos que se han estudiado en las mujeres posmenopáusicas, o por lo menos en todas las poblaciones alrededor del mundo, y que definitivamente el cáncer de aparición temprana, ¿cierto? Amerita otras hipótesis que todavía debemos estar muy conscientes de eso, y que precisamente el biobanco de sueros y de genética yo creo que van a dar, es un tesoro para poder contestar las preguntas en esa población.
Una de las preguntas que tenemos aquí tiene que ver con, digamos, las diferencias que se observan de acuerdo a los subtipos. Entonces, digamos, específicamente, aquí es, bueno, de los alimentos ultraprocesados, pero de igual forma, por ejemplo, para un factor que consideramos como un factor reproductivo de relevancia, que es lactancia. Entonces, lactancia en los CR positivos parece que previene, mientras que en los CR negativos aumenta el riesgo. Esto se ha observado en otros estudios, por ejemplo, en el Black Women's Study es lo mismo. Entonces, si nos puede dar un poquito de, pues, o sea, cuando uno ya los está viendo, digamos, realmente, ¿qué queremos decir biológicamente con R positivo, R negativo, y cuál es la expectativa respecto a su diferencia en factores de riesgo?
Well, this is also something that we should really better understand, because triple negative breast cancer are something that is, as I said, some risk factors are really different for triple negative breast cancers. The fact of having a higher percentage of triple negative breast cancer in younger women, that is complicating things a bit more, but definitely I think this is what we really need to do, and we need to have these kind of studies in young women and well phenotyped cancer, so that we can really target and do something to better understand the factors for, especially for triple negative breast cancer, or estrogen receptor negative, let's say, because these are the cancers where the mortality is really higher, because therapies, they do not really work very well. So I think that's the really important point that must be studied even more.
So, biológicamente, pues, como que todavía es entender qué significa un R un ER negativo es un R positivo? Digo, porque en el caso de lactancia como que sí llama mucho la atención, porque no, o sea, los estimadores sí son muy, son como si fuera para lo completamente en la dirección opuesta. Entonces, o sea, lo que digas es que biológicamente realmente no entendemos.
Pues bueno, eso es lo que tenemos de conocimiento precisamente con los tumores R positivos, ¿no? El papel tan importante del estrógeno y ahí la pregunta es, ¿qué es los factores que modulan los niveles de estrógeno? Porque en los otros, lo que nosotros entendemos es que precisamente no se expresan, ¿no? Esos receptores. Ninguno de los otros dos o de los otros tres que han sido está identificados en la fecha. Y la pregunta es, ¿qué modulan esos niveles? Pero yo diría también, no solamente desde el punto de vista del estilo de vida, sino también de los puntos de vista de mutaciones somáticas, y de ahí que la ancestría, por ejemplo, se le haya visto tanto importancia y que explique en cierto grado las diferencias a veces en los riesgos en cáncer de mama. Ahora, hay una dificultad grande y es cómo medir ancestría y toda la relación que hay con los factores socioeconómicos y, por supuesto, estilos de vida en nuestros continentes, ¿no? Entonces, ahí donde verdaderamente yo diría que estudios también diseñados donde emparejamos inclusive por lugar de residencia, que estaríamos ajustando por el nivel socioeconómico, nos pueden ayudar precisamente a contestarnos esas preguntas y valdría la pena este diseño replicarlo y seguirlo haciendo y amplificarlo inclusive a las mujeres posmenopáusicas y a la sobrevida para entender mucho más de este panorama tan intrincado que tiene el tema de cáncer de mama.
Pues bueno, pues ya estamos en tiempo. Les agradezco enormemente. Digo, perdón, Sabina, estamos un poquito en tiempo. Gracias, Sabina. Gracias, Gloria. Gracias a todo el mundo por su participación. Muchas gracias a todo el equipo, a la doctora Leticia Torres. Y bueno, pues seguimos adelante con este ciclo de seminarios. Y bueno, pues estamos muy interesados en seguir pensando cómo podemos utilizar una plataforma de investigación que ya está desarrollada, que ya existe. Hay muchas oportunidades, creo yo, entiendo, de colaboración con la YARC y de colaboración con el estudio PRECAMA. Nosotros tenemos dentro de nuestro instituto una excelente colaboradora y entonces creo que es importante nada más subrayar que este recurso existe y está disponible y seguirá aportando enormemente para nuestro entender, digamos, de esta enfermedad. Muchísimas gracias y bueno, pues estamos en comunicación. Gracias, Sabina. Gracias, Gloria.
Muchísimas
Gracias a
gracias a
todo el
vosotras.
mundo.
Muchísimas gracias al instituto por preocuparse por estos
Muchísimas
temas.
gracias.
Muchas gracias.
Hasta luego.
Hasta luego.
Rodrigo.